An experiment is a procedure carried out to verify, refute, or validate a hypothesis. Experiments provide insight into cause and effect by demonstrating what outcome occurs when a particular factor is manipulated. Experiments vary greatly in goal and scale, but always rely on repeatable procedure and logical analysis of the results. There also exist natural experimental studies. A child may carry out basic experiments to understand gravity, while teams of scientists may take years of systematic investigation to advance their understanding of a phenomenon. Experiments and other types of hands-on activities are very important to student learning in the science classroom. Experiments can raise test scores and help a student become more engaged and interested in the material they are learning, especially when used over time. Experiments can vary from personal and informal natural comparisons to highly controlled uses of experiments vary considerably between the natural and human sciences. Experiments typically include controls, which are designed to minimize the effects of variables other than the single independent variable. This increases the reliability of the results, often through a comparison between control measurements and the other measurements. Scientific controls are a part of the scientific method. Ideally, all variables in an experiment are controlled and none are uncontrolled. In such an experiment, if all controls work as expected, it is possible to conclude that the experiment works as intended, and that results are due to the effect of the tested variable. Overview. In the scientific method, an experiment is an empirical procedure that arbitrates between competing models or hypotheses. Researchers also use experimentation to test existing theories or new hypotheses to support or disprove them. An experiment usually tests a hypothesis, which is an expectation about how a particular process or phenomenon works. However, an experiment may also aim to answer a what-if question, without a specific expectation about what the experiment reveals, or to confirm prior results. If an experiment is carefully conducted, the results usually either support or disprove the hypothesis. According to some philosophies of science, an experiment can never prove a hypothesis, it can only add support. Similarly, an experiment that provides a counterexample can disprove a theory or hypothesis. An experiment must also control the possible confounding factors, any factors that would mar the accuracy or repeatability of the experiment or the ability to interpret the results. Confounding is commonly eliminated through scientific controls or, in randomized experiments, through random assignment. In engineering and the physical sciences, experiments are a primary component of the scientific method. They are used to test theories and hypotheses about how physical processes work under particular conditions. Typically, experiments in these fields focus on replication of identical procedures in hopes of producing identical results in each replication. Random assignment is uncommon. In medicine and the social sciences, the prevalence of experimental research varies widely across disciplines. When used, however, experiments typically follow the form of the clinical trial, where experimental units are randomly assigned to a treatment or control condition where one or more outcomes are assessed. In contrast to norms in the physical sciences, the focus is typically on the average treatment effect or another test statistic produced by the experiment. A single study typically does not involve replications of the experiment, but separate studies may be aggregated through systematic review and meta-analysis. Of course, these differences between experimental practice in each of the branches of science have exceptions. For example, agricultural research frequently uses randomized experiments. Similarly, experimental economics often involves experimental tests of theorized human behaviors without relying on random assignment of individuals to treatment and control conditions. History. Francis Bacon, an English philosopher and scientist active in the 17th century, became an early and influential supporter of experimental science. He disagreed with the method of answering scientific questions by deduction and described it as follows. Having first determined the question according to his will, man then resorts to experience, and bending her to conformity with his placets, leads her about like a captive in a procession. Bacon wanted a method that relied on repeatable observations, or experiments. Notably, he first ordered the scientific method as we understand it today. There remains simple experience, which, if taken as it comes, is called accident, if sought for, experiment. The true method of experience first lights the candle, hypothesis, and then by means of the candle shows the way, arranges and delimits the experiment, commencing as it does with experience duly ordered and digested, not bungling or erratic, and from it deducing axioms, theories, and from established axioms again new experiments. In the centuries that followed, people who applied the scientific method in different areas made important advances and discoveries. For example, Galileo Galilei accurately measured time and experimented to make accurate measurements and conclusions about the speed of a falling body. Antoine Lavoisier, a French chemist, used experiment to describe new areas, such as combustion and biochemistry, and to develop the theory of conservation of mass. Louis Pasteur used the scientific method to disprove the prevailing theory of spontaneous generation and to develop the germ theory of disease. Because of the importance of controlling potentially confounding variables, the use of well-designed laboratory experiments is preferred when possible. A considerable amount of progress on the design and analysis of experiments occurred in the early 20th century, with contributions from statisticians such as Ronald Fisher, Jerzy Neyman, Oscar Kempthorne, Gertrude Mary Cox, and William Gemmell Cochrane, among others. This early work has largely been synthesized under the label of the Rubin causal model, which formalizes earlier statistical approaches to the analysis of experiments. Types of experiment. Experiments might be categorized according to a number of dimensions, depending upon professional norms and standards in different fields of study. In some disciplines, a true experiment is a method of social research in which there are two kinds of variables. The independent variable is manipulated by the experimenter, and the dependent variable is measured. The signifying characteristic of a true experiment is that it randomly allocates the subjects to neutralize experimenter bias, and ensures, over a large number of iterations of the experiment, that it controls for all confounding factors. Controlled experiments. A controlled experiment often compares the results obtained from experimental samples against control samples, which are practically identical to the experimental sample except for the one aspect whose effect is being tested. A good example would be a drug trial. The sample or group receiving the drug would be the experimental group, and the one receiving the placebo or regular treatment would be the control. 1. In many laboratory experiments it is good practice to have several replicate samples for the test being performed and have both a positive control and a negative control. The results from replicate samples can often be averaged, or if one of the replicates is obviously inconsistent with the results from the other samples, it can be discarded as being the result of an experimental error. Most often, tests are done in duplicate or triplicate. A positive control is a procedure similar to the actual experimental test but is known from previous experience to give a positive result. A negative control is known to give a negative result. The positive control confirms that the basic conditions of the experiment were able to produce a positive result, even if none of the actual experimental samples produce a positive result. The negative control demonstrates the baseline result obtained when a test does not produce a measurable positive result. Most often the value of the negative control is treated as a background value to subtract from the test sample results. Sometimes the positive control takes the quadrant of a standard curve. An example that is often used in teaching laboratories is a controlled protein assay. Students might be given a fluid sample containing an unknown amount of protein. It is their job to correctly perform a controlled experiment in which they determine the concentration of protein in fluid sample. The teaching lab would be equipped with a protein standard solution with a known protein concentration. Students could make several positive control samples containing various dilutions of the protein standard. Negative control samples would contain all of the reagents for the protein assay but no protein. In this example, all samples are performed in duplicate. The assay is a colorimetric assay in which a spectrophotometer can measure the amount of protein in samples by detecting a colored complex formed by the interaction of protein molecules and molecules of an added dye. In the illustration, the results for the diluted test samples can be compared to the results of the standard curve to estimate the amount of protein in the unknown sample. Controlled experiments can be performed when it is difficult to exactly control all the conditions in an experiment. In this case, the experiment begins by creating two or more sample groups that are probabilistically equivalent, which means that measurements of traits should be similar among the groups and that the groups should respond in the same manner if given the same treatment. This equivalency is determined by statistical methods that take into account the amount of variation between individuals and the number of individuals in each group. In fields such as microbiology and chemistry, where there is very little variation between individuals and the group size is easily in the millions, these statistical methods are often bypassed and simply splitting a solution into equal parts is assumed to produce identical sample groups. Once equivalent groups have been formed, the experimenter tries to treat them identically except for the one variable that he or she wishes to isolate. Human experimentation requires special safeguards against outside variables such as the placebo effect. Such experiments are generally double-blind, meaning that neither the volunteer nor the researcher knows which individuals are in the control group or the experimental group until after all of the data have been collected. This ensures that any effects on the volunteer are due to the treatment itself and are not a response to the knowledge that he is being treated. In human experiments, researchers may give a subject a stimulus that the subject responds to. The goal of the experiment is to measure the response to the stimulus by a test method. In the design of experiments, two or more treatments are applied to estimate the difference between the mean responses for the treatments. For example, an experiment on baking bread could estimate the difference in the responses associated with quantitative variables, such as the ratio of water to flour, and with qualitative variables, such as strains of yeast. Experimentation is the step in the scientific method that helps people decide between two or more competing explanations, or hypotheses. These hypotheses suggest reasons to explain a phenomenon, or predict the results of an action. An example might be the hypothesis that, if I release this ball, it will fall to the floor. This suggestion can then be tested by carrying out the experiment of letting go of the ball, and observing the results. Formally, a hypothesis is compared against its opposite or null hypothesis. The null hypothesis is that there is no explanation or predictive power of the phenomenon through the reasoning that is being investigated. Once hypotheses are defined, an experiment can be carried out and the results analyzed to confirm, refute, or define the accuracy of the hypotheses. Natural experiments. The term experiment usually implies a controlled experiment, but sometimes controlled experiments are prohibitively difficult or impossible. In this case, researchers resort to natural experiments or quasi-experiments. Natural experiments rely solely on observations of the variables of the system under study, rather than manipulation of just one or a few variables as occurs in controlled experiments. To the degree possible, they attempt to collect data for the system in such a way that contribution from all variables can be determined, and where the effects of variation in certain variables remain approximately constant so that the effects of other variables can be discerned. The degree to which this is possible depends on the observed correlation between explanatory variables in the observed data. When these variables are not well correlated, natural experiments can approach the power of controlled experiments. Usually, however, there is some correlation between these variables, which reduces the reliability of natural experiments relative to what could be concluded if a controlled experiment were performed. Also, because natural experiments usually take place in uncontrolled environments, variables from undetected sources are neither measured nor held constant, and these may produce illusory correlations in variables under study. Much research in several important science disciplines, including economics, political science, geology, paleontology, ecology, meteorology, and astronomy, relies on quasi-experiments. For example, in astronomy it is clearly impossible, when testing the hypothesis, suns are collapsed clouds of hydrogen, to start out with a giant cloud of hydrogen, and then perform the experiment of waiting a few billion years for it to form a sun. However, by observing various clouds of hydrogen in various states of collapse, and other implications of the hypothesis, we can collect data we require to support the hypothesis. An early example of this type of experiment was the first verification in the 17th century that light does not travel from place to place instantaneously, but instead has a measurable speed. Observation of the appearance of the moons of Jupiter were slightly delayed when Jupiter was farther from Earth, as opposed to when Jupiter was closer to Earth, and this phenomenon was used to demonstrate that the difference in the time of appearance of the moons was consistent with a measurable speed. Field experiments are so named to distinguish them from laboratory experiments, which enforce scientific control by testing a hypothesis in the artificial and highly controlled setting of a laboratory. Often used in the social sciences, and especially in economic analyses of education and health interventions. Field experiments have the advantage that outcomes are observed in a natural setting rather than in a contrived laboratory environment. For this reason, field experiments are sometimes seen as having higher external validity than laboratory experiments. However, like natural experiments, field experiments suffer from the possibility of contamination. Experimental conditions can be controlled with more precision and certainty in the lab, yet some phenomena cannot be easily studied in a laboratory.